Next, we have a last lesson of the circulatory system, grouping of blood. Lesson 25, grouping of blood, and after that, the Rhesus system. Again, something related to the grouping of blood. If you want to understand the grouping of blood, we need a background knowledge of antigen and antibodies. We have heard these names many times, antigen and antibodies, but we want to understand these two terms if we want to understand the grouping of the blood. Okay, now we try to understand what is antigen. Right. Now here, this box represent the human immune system. Here, same thing, human immune system. Now, what is the function of human immune system? Human immune system protect us from harmful things. It protect us from harmful things. If this thing can damage our body, it could be a live thing. It could be just a molecules. It could be a live thing like a bacteria, virus, or non-living molecules, just the molecules, any kind of molecules. So basically, anything which trying to damage our body, which is going to harm our body, immune system try to protect by fight with this harmful thing. Immune system going to fight with the harmful thing, and this is going to completely remove from the body. If this is going to harm and it's going to remove from our body by the immune system. Let's say this represent red color dot represent a thing which can be harm to the body. Now, in the first case, if the immune system activate, so this blue color spiral type of arrows represent activation. Now, in this case, the second instance, there is no activation. That means this causes the activation. This is the cause and this is the effect. And in this case, it doesn't cause immune system activation. So there's no cause and no activation. So we can write here now this is a antigen. This is not an antigen. So by looking at the picture, we can define antigen. So we can define anything which can cause activation of the immune system is called antigen. If things which is not activation, which is not activate the immune system, they are not antigen. So antigens are molecules which can activate immune system. Now this activation and effect. So activation and effect, we're going to learn in a bigger way in immunity. What is the significance of activation? How do we know that immune system activate? How do we know that the immune system activation at which level? And how we can measure it? How we can measure activation of the immune system? So we can find many different activations of the immune system. I mean, it's a complex process and it has many parts. One component of activation is production of a special group of protein called antibodies. Antibodies. Because this antibody helps to fight with this antigen, helps to remove the antigen. Antibodies help to remove the antigen. So antigen could be harmful. This harmful effect is removed. One component is antibody. Out of several components, antibody is one component which is going to remove this antigen. Okay, second one, we don't want to discuss anything because it is not harmful. Right? For example, if we take a chemical into our body, this chemical is not part of our body. The chemical is not part of our body. Basically, the immune system is designed to identify molecules which is not part of a person body. The molecules which is not part of person's body. So any molecules which is not part of the body is going to identify and fight with it. But you know, some molecules are not part of our body. So if you take cow milk, some of the proteins, some of the things might not uh, match with our body. If you take animal protein as food, fish, meat, some of the proteins are not related to our body. But generally, we know that if we take this kind of protein, body will not usually not make a reaction. If you take a medicine, a drug, this drug is going to the body. This drug is a chemical, different chemical. It is not a part of the body, but immune system not activate. So in this case, they are not considered as an antigen because immune system try to find a molecule which is not a part of the body, and secondly, immune system identify whether this molecule is harmful or not. If both matching, then the immune system activate. Activation is a big process. It's a big process, activation or effect. But part of that kind of molecules produce antibodies. Antibodies helps to remove this antigen which is harmful to the body. So we should learn this kind of a basic knowledge in immunology, immunity, but understand the blood group, we also need certain level of immunity. So antigens are mostly proteins. So they are mostly glycoproteins. It doesn't mean other molecules cannot be antigen, but most of the proteins are glycoproteins, and they are not a part of the body, and they could harmful to our body. So to become antigen, to become a candidate of antigen, it need a certain requirement. The requirements are it should be harmful, and it should be a foreign foreign molecules coming from outside. It should be harmful. It should be mostly the glycoproteins. To become antigen, there should be several criteria fulfilled, right? So these criteria are, one is it should be foreign, it should be harmful to the body, and it should be a glycoproteins. So this is a general characteristic to become antigen. So immune system now activate to produce different components to remove this antigen. One component is antibody. Okay, we're going to learn more detail about this concept in immunity, but just to understand the blood group, we're going to learn antigen and antibody. I hope now you have a small idea about antigen and antibody. Now, which component responsible to form blood group? Because blood has plasma, blood has erythrocyte, blood has white blood cells, blood has platelets. So which component is responsible to make the blood group? Now, understanding this, if a person donate blood to another person, this person blood to another person. Now this person has immune system. Now immune system think if something from this person, it has a foreign, it is harmful, and it could be a glycoprotein. So then molecules are not in a person, blue color person is not in the body. An immune system think it is harmful situation, and these molecules mostly are glycoproteins. Then as we learn what is the outcome, the person immune system activate against this kind of a things coming from the person colored with red. If blood has anything which is foreign, which could be harmful, which could be glycoprotein like things, then the person immune system activate to remove these things. Otherwise, if everything is same in human, like the blood exactly same, there's no difference in the blood of person A and B, then we don't want to talk about the groups. There's no groups, it's just one. So then there should be certain different molecule existing in the blood. Now, main component, main player, main molecules of blood, which is the foundation to make, is coming from red blood cell. Or erythrocyte. Erythrocyte or red blood cells. So this is the phospholipid layer, and this is a phospholipid bilayer. And we have learned in cell membrane, there are many proteins embedded like this. And this protein also combined with carbohydrate to make glycoproteins like this. So this is the cell membrane and the proteins. Now, there are two type of proteins. One is glycoprotein A, other one is glycoprotein B. So there are two different types. So they are different. A and B are different. A is a one type of glycoprotein, B is a one type of glycoproteins. Then in human, has B proteins on red blood cells, and some has A. So based on this is a red blood cell, this is a red blood cell, this is a red blood cell, and this is A, This is A, and this is a B. This is a B. Now, some people they have A glycoprotein on red blood cell membrane. Some people they have a B glycoprotein on red blood cell membrane. Some people they have a both A and B glycoproteins. And another people, group of people, so they don't have A glycoprotein, they don't have B glycoproteins. Now you can understand in human population, some people have A glycoprotein on red blood cell. Some people they have a B glycoprotein on red blood cells. Some people have A and B both glycoprotein on cell membrane of red blood cell. And some group of people they don't have any type of glycoprotein on red blood cell membrane. As a result, we basically divide human blood into four groups: Group A, Group B, Group AB, and when there's nothing, we call it O. So these are the four blood group in human: blood group A, blood group B, blood group AB, and blood group O. So there are four blood groups among human, four main blood groups among human. So we can talk about different blood groups, but here this is the main type of blood groups: AB and O. Okay. Now, going further, let's take blood group A. Blood group A, red blood cell membrane, that's a phospholipid membrane, and uh, this is the glycoproteins. So they have A, glycoprotein A. Group A. So let's name this again: red blood cells, glycoprotein A. Now this is going to represent the plasma part of the blood. This is the plasma. Now coming to the antibody story. When it comes to the antibody story, there are many different antibodies in the plasma. So these antibodies are formed due to the exposure to different antigens in the life. Antibodies are formed. If we expose to a disease, then antibodies form, antibodies stay in the blood. If we expose to a dengue fever, dengue virus, antibodies are formed against dengue virus, antibodies stay in the body. If we expose to COVID-19, antibodies are formed, antibodies stay in the body. So if we expose to many different antigen, and antibodies are stay in the body. Some antibodies stay longer, some antibodies stay shorter. Now there is a group of antibodies in these people, blood group A. So these antibodies are called antibody B. Antibody B. So antibody B has no effect on antigen A because there are two different things. Antigen A is antigen A. Antibody B is something different. It doesn't match with antigen A. There's no matching. That means they have no interactions. So this antibody and antigen has no interaction. It just stays in the plasma. Right. So they have antigen A. These people have antigen A. And these people have antibody B. Antigen A, antibody B. So there's no interaction between them because they cannot recognize each other, and there's no binding, there is no reactions between them. So this is the blood group A. They have antigen A on the red blood cells, and plasma has antibody B. Now let's take a person with blood group B. That's a red blood cell. This could be the plasma membrane, cell membrane, and this is the antigen glycoproteins. So it should be B. And this represent the plasma. Plasma has antibody. This antibody is A. The type of antibody is A. Again, there is no interaction between each other. This is a B uh, antigen, and this is the A antibody. So there is no interaction between each other. Now let's consider there is a exchange of blood, which is called transfusion, blood transfusion, donation of blood happen between A and B. It could happen like this. So this is a red blood cell. This is a red blood cell. I just got a new slide. This is a red blood cell, and this is a red blood cell. And this is the membrane. This is again cell membrane. And these are the antigen. And this represent the plasma part. This is again the plasma. Okay, so this is a person A blood group, this person B blood group. So A has a A antigen, B has a B antigen on the surface of red blood cells. And now coming to the antibodies. So what are the antibodies we can find here? Here it's a B and here's a A. Okay, let's assume we give a person A blood to person B. Now, if we give A blood to B, or we can give other way, B to A. In this case, what we're going to observe, if A blood is going to be, this represent the red blood cell of A, and this represent antibody of A. So this is a blood of A now, right? This is a blood of A. Now it's mixed in the B. Blood mixed together, both in A and B. Now what is going to happen in this case? the antigen in person A the type of antigen in person A is uh, A antigen. Now, person blood group B has A antibody. Now, antibody A can recognize A antigen. This is a good recognition. The antibody A can recognize A antigen. This is antibody A and this is antigen. Now, what will happen now if it recognized? Then start the process of removing, destroying, thinking this is harmful to the body. So this is a perfect match. B has A antibody. When blood coming from A has A antigen on the red blood cells. So this is perfect match, can identify. Now try to make some kind of a reaction. So this is not possible, this combination. Now same thing happen if you go to B to A. So if it goes to B to A, this is the B red blood cell. And B has D antigen and A has B antibody. Now this can be recognized again and make a response. So this is also not possible then. It's not matching because antigen antibodies are recognized and then immune system trying to make reactions for this kind of matching and transfusion or donation of blood. Okay. We are taking now AB blood group. This is a AB red blood cell and this is the cell membrane. And this person has both A and B. And this is the plasma. So A, B, A, B, A, B, A, B. And there's no antibodies. So AB has no antibodies in the plasma. It has only the antigen AB blood group. Now AB blood group if AB blood group get blood group A or B, let's see what's going to happen. This is a B, this is a A. Now A plasma has B antibody, B plasma has A antibody. This is A antibody, this is B antibody. Now, if this blood come to AB, there is no antibodies in AB site to recognize A antigen. That means this is B antigen, this is A antigen. This is B antigen, this is A antigen. Now this is possible, and this is also possible. Because if A blood is mixing with AB, AB has no antibodies, no recognition. If B comes, no recognitions. AB can't recognize because they don't have antibodies. Let's see what will happen the other way now. AB is giving blood to B. If AB is giving blood to B, B has A antibody, can recognize. This is not possible. Now AB is giving blood to A now. So B antibody is there and AB has B antigen. That can be also recognized, not possible. So donation is possible, A to AB, B to AB. Not AB cannot give blood to A or B. Okay. Next, we go to a O blood group. Cell membrane. And this is the plasma. And they have both antibodies A and antibody B. Antibody A and B in the plasma. Okay, now let's see if we give to A, what will happen? To AB, what will happen? And to B. A, B, AB. Right. Now, A has A antigen, B has B antigen, AB has both. Plasma, A has B antibody, B has A antibody, and AB has no antibody in the plasma. This is A antibody, this is a B antibody, and there's no antibody. So A, and this is a B. Right, now O, A, B, AB. O is given to A, donating blood to A. So A has A antibody, but there's no antigen to recognize. O has no antigen to recognize. O red blood cell has no antigen, then it's not recognized. This is okay. O is given to B. B has B antibody. B antibody cannot recognize anything because no antigen on A, on O blood group. This is also possible. So O is given to AB. So A has no antibodies, nothing will recognize, nothing will happen. So this is also possible. So it is always possible to give blood to the same blood group, that A can give to A, B can give to B, AB can give to AB, O can give to O. This is always possible because same blood group. So we don't want to find why it is that, because same blood group, nothing will happen. Nothing will happen with blood transfusion of the same group. Now O can donate blood to A, B, AB and O. So O is a universal donor that can give to anyone. Universal donor. Now here we have seen AB can accept from A, AB can accept from B, and AB can accept from O. So AB is a universal recipient. So recipient is the one who is taking the blood, one is giving is a donor. AB is a universal recipient. O is a universal donor. So we have four blood groups, AB, O, and AB. And these blood groups are decided by the antigen on the surface of the red blood cells. Now this antigen of red blood cells, which decide the blood group, that is the A or B, is considered, these antigens are called agglutinogen. Agglutinogen. So A and B are antigens, and when they are in the red blood cells, they get a name called agglutinogen. So blood cell antigens are called agglutinogen. They are on the red blood cells, they are glycoproteins. Okay, here we have learned now A has A agglutinogen, B has B agglutinogen, AB has both agglutinogen, A and B, and O has no agglutinogen. When it comes to the plasma, A has B antibodies, B has A antibodies, and AB has no antibodies, and O has both antibodies in the plasma, A and B. So this is the end of the blood grouping, and we have to find another type of group called Rhesus blood grouping.